Understanding the aging process is a frontier in science today. Aging affects every individual on the planet, yet the precise mechanisms are not yet fully understood. The concept that aging is not inevitable is evidenced by several multicellular taxa that do not exhibit age-related increases in mortality. Thus, it appears that aging is neither evolutionarily inevitable nor impossible to overcome at the mechanistic level. However, to overcome aging, it must first be understood. Cause what even is aging anyway? There is a list of 9 hallmarks of aging. Shortening of telomeres has long been considered one of the main causes. An accumulation of senescent cells, which are zombie-like cells. There is poor intercellular communication. There's mitochondrial dysfunction and genomic instability. These are examples of the 9 different features of older cells. But the question is, are they the cause of aging, or are they the result of a deeper root cause? This root cause was previously thought to be damage to our DNA that accumulates over the course of our lives. But this is refuted by the fact that you can take an adult cell and clone it into a new organism that will live just as long as an uncloned organism of the same species. This suggests that we do not lose information in our DNA. A new hypothesis proposes that we do lose information, but not in our DNA or genome, but in our epigenome. What is this epigenome? Every cell in your body has the same DNA, the 23 pairs of chromosomes, but the epigenome determines which genes are used in every cell. It is a very complex collection of proteins and modifications that package and alter the DNA in a certain way so that only a certain set of genes are accessible for use, while other genes are packed away and cannot be used. Thus, it is the epigenome that causes cells to differentiate, for example into a brain cell or a liver cell, by turning on only the DNA associated with that particular tissue. So this information theory of aging suggests that as we get older, we lose information in the epigenome and therefore cells lose their identity, leading to aging. So over time, histones are no longer returned to the right places and DNA methylation is added in places where it shouldn't be. DNA methylation is simply adding a methyl group to specific parts of the DNA, which changes the activity of a DNA segment. This DNA methylation pattern can now be analyzed in the lab. By taking samples from multiple tissues and analyzing the methylation status of specific methylation sites, called CpGs, which are either methylated or not, and applying a so-called epigenetic clock algorithm to them, we can predict your biological age. A DNA methylation-based age estimator like this, such as the Horvath clock, can accurately predict biological age across the entire human lifespan. The algorithm was initially developed using machine learning and a penalized regression model on publicly available DNA methylation data. The optimization algorithm selected 353 CpGs by minimizing the error associated with estimating chronological age. Measuring the DNA methylation status of this collection of CpGs, with each one either being methylated or unmethylated, is the most accurate method to molecularly measure chronological age to date. The clock revealed that the rate is fast in the developmental stages of life and reaches a slower constant during the maintenance part of life. In this way, we can easily tell whether a person is biologically older than their calendar age, represented by the yellow area, or biologically younger, represented by the green area. With these epigenetic clocks, we can easily test the effects of certain environmental factors on our epigenetic age. In this way, it has already been proven that we can take certain steps to slow down the aging process. Billions of years ago, early bacteria made an important evolutionary step. They evolved 2 distinct ways of living. When times were favorable, they used their energy to grow and reproduce. But when times were tough, they used their energy to protect and repair their cells. Both lifestyles were associated with the activation or inactivation of a specific group of genes. These longevity genes, which among other things repair and maintain the epigenome, are also found in us. How can we change our lifestyle to activate these different types of longevity genes? In summary, there are several things you can do right now to slow down the rate of your aging. Number 1. Eat less, caloric restriction. Number 2. Eat less protein, which deactivates mTOR, a type of longevity gene. Number 3. Avoid DNA damage, for example, avoid ionizing radiation, UV light and smoking. Number 4. Do some high intensity interval training, making your body think you're running from a bear or something. And number 5. Be uncomfortably hot or cold. There is still much to unravel about the aging process, making it a pioneering topic in science, and the causal impact of epigenetics on aging needs further investigation. I hope you enjoyed the video and I see you next time.